Hello everybody, and spinning disk remote focusing microscopy. It's basically work that I should say we have been working on for about 2 years at the University of Exeter. And actually you can see the next slide because I think it's not responding.
Okay, are you sharing your PowerPoint presentation as the window itself, or are you sharing your screen?
I'm showing my screen.
Okay. So if you click next, what happens? Okay, that's working now. Perfect.
So, and the need we need to answer is that of high-speed 3D imaging with high-speed intending higher than 10 Hertz volumetric imaging speed, and confined to relevant biological volumes. So we're talking between 10 to hundreds of micrometers of depth. And we know that to achieve that, well, to achieve high speed, we need at least scientific cameras that have high frame per second counts. We want confocality. There are many physical methods, but we chose to go with the structural illumination method, so the spinning disk. And to get a high refocusing speed, while using objective scanning, which, well, until recent inventions in single objective light sheet microscopy, was the one of the best methods to get high numerical aperture imaging. We used the remote focusing to avoid the problem of perturbing the sample during movement. So basically, short as possible, the sample is reproduced optically through a relay system, which is basically made of two opposing microscopes with almost unity magnification. A conjugate image is created on the other side, and the actual ray imaging objective is used to image that hologram instead of the sample. The main and most strict requirement of this strategy is that the relay system needs to have a magnification as close as possible to the ratio between the refractive indices of the two media at the sides of the relay system. So this is actually the system a bit more in detail. The LED light content is, well, we are using a spinning disk unit, the Clarity from Orox, obviously, and the light of the LED is focused onto the spinning disk. I won't go too much in detail. Last session, Filippa described it really well. So half of the light is transmitted by a pattern on the spinning disk. This pattern is focused on the desired height or depth inside the sample thanks to the remote focusing unit. And then the patterned pattern emitted light from the fluorescence is captured and refocused onto the spinning disk by retracing back the remote focusing unit. At that point, the spinning disk basically divides the light in focus plus half out focus and half out focus light, and directs these two elements onto two separate halves of the camera sensor, where they are processed and they form, they're processed to form the confocal image. And the system illumination, the camera, and mostly the spinning disk work, they can be easily controlled by micromanager, so that will be quite straightforward. But only one last issue is remaining, and it is to synchronize the movement of the objective with the acquisition of the camera. And for that, we used a data acquisition card. Until, well, into 2019, we got our first results. We use a tipping objective, even though given the geometry of the system, which basically is a commercial stand, an Olympus stand, we usually use a cover slip sample, but even that, we started with a deep-in objective with medium-high numerical aperture, which was, and we got a refocusing unit which was capable of moving between 400 micrometer range. And we got a lateral resolution of 419 nanometers with regular blue light, so 488 nanometers, and an axial resolution of 3.2 micrometers. On the right, you see the evolution of the resolutions, both lateral and axial, depending on the position of the ray imaging objective, so the third one. And you see that the spinning disk unit doesn't change anything. It doesn't introduce any problem into the resolution. So we added a simple acquisition control. Basically, the LED light and the camera acquisition just have to wait for the objective to reach its intended position. And we started the observation of live sample with this quite convenient larva of sea worm called Platineris dumerili. And at 2 days post-fertilization, this larva basically shows itself as a sphere of a few hundred cells, most of which are confined on the upper, on the top of the sphere, where they form the neural plexus, while another region of interest is the equatorial cilial band that allows this small larva to move. At 3 days post-fertilization, these larvae actually become elongated and start segmenting, and they lose the ability to move thanks to the cilia, but they develop sort of fins, the chytae, which make them able to move. And they developed a spinal cord in the posterior region. With this sample, we were able to see, kind of slowly, 1 volume per second, a volume of 24 micrometers divided in 5 planes. And yes, even with that, we were able still to catch some calcium spikes in the photoreceptors with a good enough contrast to be able to recognize them from the background. And we were also able to get some, let's say, aesthetical results. So an indication of a good image quality on the here. Unfortunately, I don't see my my mouse, but on the bottom left, you can see the 2-day larva, 2-day-old larva, and in red the photoreceptors, and in white actually the nuclei of the cells, and on the bottom right the always in red the photoreceptors, the nuclear the neural plexus, and a little bit smaller but still visible the spinal cord, and in green the almost vestigial now ciliary bands. Is it going on?
It doesn't seem like your slide is.
Okay.
Oh, there we go. Okay. Okay.
And from then, in 2020, we tried to solve some problem and increase the capabilities of the system. And when I'm talking about problems, basically, it might seem minor, but there was the magnification of the relay system wasn't as close to 1.33 since it was a water-to-air relay. It still wasn't close enough to the 1.33 magnification. So just changing what seems like a little bit, 5 millimeters, the focal length of the second tube length, we were actually able to move the range from plus minus 70 micrometers to plus minus 100 micrometers. So minor correction actually brought visible changes. And then, well, we used the cover slip corrected objective with higher numerical aperture, and we brought ourselves to 300 nanometers and 1.30 micrometers in axial resolution. And then, well, we moved, I'm waiting for the slide to, to load. We moved to achieve higher imaging speeds. always if the system allows it.
Don't worry,
Here
we
it
have
is.
there we go.
And where the main bottleneck was given by the fact that the translator mount of the ray imaging objective was actually performing its own closed loop control of the objective position, and that was making us looking was losing time, and it was also slowing down the movement of the objective. So we basically just switched off this closed loop control. We moved to open loop movement acquisition, and in order to still be able to define precisely the position of the objective, we preceded each acquisition with an adaptive correction of the of the input drive signal, basically giving the input drive signal, checking the positions actually covered by the objective, and then performing a correction on the input, and so on and so forth. To that we added, well, instead of acquiring positions at single at point where the objective stopped, we started to we decided to basically acquire images while the objective was running, provided it was not running too fast. And with this combination of strategies, we just managed to reach on the same volumes of 24 micrometers, though with 3 planes, we reached the acquisition of 12 hertz, so 12 volumes per second, even though with 5 planes it's closer to 10.7 hertz. And just to provide some examples, here is a 2-day-old larva. I'm going to show you a video of this larva behaving. So just know that the fluorescence here comes from GCamp. So we are looking at the activity of the photoreceptors. And we are using a 50 millisecond exposure time, which means a 20 hertz frequency. So I'm not seeing any movement. You are seeing something?
We see a little bit of movement, Michele, but what we could do is, if you provide us with the video file, we might be able to play it directly from here a bit later, if it doesn't show up to everybody. So yeah, let us know where that video is, and maybe Lee can load it up so that people can look at it at another part of your another part of the afternoon. Are you able to do that, Lee?
Okay. I cannot do that immediately.
But I'll need some. Ideally, the only way I can do it is if it's on something like a YouTube link. I can run it from there. But
that will be difficult. I don't have a YouTube account.
Okay, no problem. All right.
But I'll be happy to share them, and eventually, if I manage to load them on Twitter, that's when I have it.
That would be brilliant. Yes, yeah, you can add it to the.
Hopefully.
Yeah, no problem. Cool.
So by using the simple standard deviation projection of the entire stack, you can see that there are hotspots of activity, and mainly these 2 ones, which I will for now give the casual names of the random names of master cell and ciliary band cells. And basically, one can see, for example, that the time tracks of their activity are almost the same. And that is actually because this master cell is supposed to directly control these ciliary band cells, which inhibit the flailing of the cilia in the equatorial band. So this is an image that we get from the simple wide field observation mode. If I've clicked for we just need to wait for the other slide. Wait just a second, we will see the one confocal images. And fully. should have finished by now. I had taken a little bit of time for questions, but
no problem. We are running early, so that's fine.
Okay, so here again in confocal mode, master cells, ciliary band cells. You can see there's still a correspondence, even though in this case here the ciliary band cells were quite dim. And it was better when observing the video, but basically it turns out we had an unexpected explosion of activity in this central circular band of cells. And from these two small arrow eyes inside this band, we could actually catch their activity. The blue ones for the lower arrow eye, the red one for the left one. And finally, these two videos were taken in only one plane, but we tested also on the system on a two-plane acquisition. The two planes divided by simply 14 microns, and the image is taken in confocal mode. Don't worry, I've clicked. These are luckily the last 3 slides. Well, I think that I should just exit from presentation mode and just Am I still connected, or?
It looks like you're still connected. You could always try and unshare and share again.
Yeah, I'd make sure that you are sharing your whole screen, to be honest. We've seen differences between the window or the whole screen at times.
There we go.
I will just show here. Sorry. This same master cell and ciliary band dynamics was explored while switching up and down, going up and down onto the sample. And in this case, even though the exposure time was 20 milliseconds, given that we were observing more frames per cycle, we actually acquired a 12.5 hertz volumetric speed. Still getting a good contrast and getting a good confocality. Basically, the signal from the master cell is almost absent, while the ciliary band cells are kind of tall and distributed over the vertical direction, because in this case the walls are vertical since we are near the equatorial band. So to finish, we developed this 10 hertz volume scan from the 1 hertz speed we started with an axial refocusing range of ± 100 microns, proper lateral and axial resolution for subcellular imaging, and we demonstrated interesting correlation between the activity of the master cell and ciliary bands in the Platinaris dumerili, which is confirmed as a good biological test sample. Finally, I would like to give my thanks to the group I've worked at the University of Exeter until this February. So the PI, Alex Corbett, and the ex-colleague Sharika Mohanan. And obviously, I'm grateful for the funding from the EPSRC body. And I would like to thank you all for your attention. And also, it is nuisance. So I need to be sorry for the connection problems. Hopefully, there will be a little bit more time for you to ask questions.
Thank you so much, Michele. Yes, we were running early anyway, so that was absolutely fine. We can take our time with the questions. A really interesting talk. Don't worry about the technical problems. There have been a few niggles along the day, so everybody has had a couple of problems. So, but the important thing is the content was very good. I've loved working with you guys at the University of Exeter to get that system set up. And if anyone's interested in hearing more about it, there is a publication and there is an application note on our website. So if you want to hear more about this remote focusing system that's working with our Clarity laser-free confocal, then do look it up or contact Michele or Alex. Yeah, good luck in your new role, Michele. Yeah, I know Alex will miss you. So we do have a couple of questions from Mika, who works at ICIT in Germany. Now, he's been working quite hard on getting high-speed acquisition running with the Clarity LFC. He's not using remote focusing, but he asks, would you be able to speed up the acquisition if you could stream the data directly to disk?
Well, all the videos you saw were already taken using direct memory access, and so we were basically going at full speed with the SATA disk that we had, and we were also using camera link connection because, well, we were using an Orca camera which provides camera link connection, which is, as far as I know, the fastest kind of connection that is available for scientific cameras, at least. And so I think that on the side of, let's say, deep software strategies or hardware strategies that we can use, we were at the best of our capabilities. Also, I need to point out that the 50 millisecond exposure time was by the way, there was the second question exactly about this. So the 50 millisecond exposure time was just let's say a convenience exposure time. In reality, with this combination of direct memory acquisition and of camera link connection, we actually saw easily images at almost 100 hertz frame acquisition this time, and we were actually trying to get a two-plane acquisition on that side. Unfortunately, still high speeds elude the extremely custom software that I developed for the application. So it's not really a technological problem, it's just an implementation problem at the moment.
Okay, so what would you say is the biggest speed bottleneck that you're facing? Is it the Z movement, or is it the camera, or is it simply getting the data onto the computer fast enough?
Well, I would say that at the moment, the speed bottleneck is not referring anymore to the hardware. As I was saying, we there's still a last step in software development that needs to happen, and that basically involves the synchronization, the precise synchronization of the between the camera and the movement of the objective. So at the moment, the system would be easily capable of acquiring 2 frames at at more than 20 hertz. 2 planes at more than 20 hertz, but there is a problem of long-term synchronization. Metamorph, I admit I've never heard about this this software. If you actually would like to share with me a few links and info, I would be glad to take them. So basically, that the bottleneck at the moment is mostly the capability of the software to respond to the high speed required.
Okay. Yeah. So
I'm sorry.
So yeah, Mika represents aurochs in the German-speaking countries and various other territories around the world, and so he's been really pushing the boundaries of what our technology can do. And recently, there's been an implementation of our system in Metamorph, which is it's quite an old software package now. It went out of fashion a little bit, but it's ever so powerful for doing this sort of very fast streaming acquisition. So I can put both of you in touch with each other, and you can have a chat offline and see what you can achieve. Yeah, great. Any more questions for Michele? Lee has kindly put a link to your application note on the chat folder. Oh, there's another question from Mika. Oh yeah, just saying what he's managed to achieve in Germany in his basement.
Impressive.
Yeah, so yeah, definitely I'll put you in touch offline, and you can see how fast you can push the system, which would be really interesting for us to hear.
Okay,
so
thank you.
Brilliant. Thank you so much, Michele. Good luck in your new venture, and I hope to catch up with you again sometime in the future.